Enzymes are essential biological molecules which are involved in chemical reactions within the cell. Many factors can affect the activity of these enzymes, such as temperature, pH, enzyme concentration, substrate concentration, and the presence of inhibitors or activators. This lab looked into how substrate concentration affected the activity of enzymes, specifically the rate at which catalase catalyzes the decomposition of varying hydrogen peroxide concentrations. Catalase is found in the liver, where it breaks down hydrogen peroxide, a cytotoxic agent, into water and oxygen. It is believed that a 0% concentration will result in no activity, and as the hydrogen peroxide concentration increased, so would the rate of reaction, leading to a greater volume of oxygen gas produced. With the higher concentrations of substrate, the enzyme would have a higher chance of binding and also have more to bind to, hence be able to release more products.
For our procedure, we made sure to have 10 milliliters of solution for all dilutions and used a baseline concentration of 3% hydrogen peroxide to make all dilutions. For the 2.4 concentrations, 8 milliliters of hydrogen peroxide was mixed with 2 milliliters of water for a total of 10 milliliters. For every greater diluted solution, 2 milliliters less of hydrogen peroxide was used and 2 milliliters more of water, until we got to 0% where the 10 milliliter solution was only water. For the catalase, we pipetted 40 microliters onto a paper disc. The baseline procedure was then followed.
The highest concentration starts off by producing the most oxygen, but then the reaction greatly slows, so that there's not as much oxygen produced after the first 30 seconds. The concentration that created the most oxygen was 1.2%. Although it started off producing less oxygen, the reaction did not drop in speed as much, allowing it to create more oxygen between 30 and 90 seconds. After the 1.2%, the next lower concentration, 0.6%, greatly drops in oxygen production. This shows that there was not enough substrate for the reaction. The 0% produced no oxygen since there is no substrate present. The main idea that should be drawn from this graph is that 1.2% appears to be the optimal concentration for the decomposition by the catalyst. An overall trend of rate of oxygen production decreasing over time is seen in the data. 3% has the highest rate, then drops down to a rate of only about 0.1 milliliters per second. Both 2.4% and 1.8% show an interesting trend of slowing down in the first half, then increasing production in the second half. 1.2% has the most steady decline, which shows what was being said before, that this is the optimal concentration. Also, adding on to the previous graph, 0.6 shows a starting rate of production of only 0.1 milliliters per second, and slows down even further to about 0.05 milliliters per second. The trend that can be observed in this graph is a rising rate of oxygen production, with again 1.2% being the concentration that has the highest rate of production. You can also see a sharp decline in the rate of oxygen production in the 0.6% concentration, and then the decline to a rate of 0. On this graph, the 2.4% is an outlier, as it does not follow the trend that can be observed with the rest of the data.
The reason these trends can be observed is because of the way enzymes work on a molecular level. They catalyze reactions by binding to the substrate. In this case, the reaction that is being catalyzed is the decomposition of hydrogen peroxide into water and oxygen gas. As the concentration of substrate increases, the higher the chance there is of a molecule of the substrate colliding with an enzyme. This effect is what creates the rise in oxygen creation between 0 and the 1.2% test. The reason there is no increase in the creation of oxygen between 1.2 and 3% concentration is because of the fact that there is a fixed amount of enzymes available, leading to enzyme saturation after a certain point. Our experimental results partially agreed with the hypothesis. We say that as concentration increased, the amount of oxygen created would also increase. Our results showed this to be true up to a point. After a 1.2% concentration, this trend changed to the amount of oxygen being created staying relatively the same. This proves what we had learned in class, that substrate concentration leads to a faster reaction until saturation. Further looking into catalase, it is said that catalase has a maximum reaction rate of 0.0144 moles per second. Our experiment showed a maximum rate of 0.32 milliliters per second, or 0.0134 moles per second. This is a percentage error of 6.9%, which could be attributed to errors in the lab, as well as the fact that the concentration that led to this highest oxygen production may not have been the exact ideal concentration. To try and keep the testing consistent throughout to avoid errors, the amount of substrate in catalase was kept consistent in our experiment, although some errors still occurred throughout the experiment. All values measuring oxygen production under 10 milliliters would have had error, as the graduated cylinder we were using had measurements starting at 10 milliliters, and so values below this amount had to be estimated. There is also instances where the catalase would start to run down the walls of the reaction chamber while the substrate was being added, which could have led to a small reaction taking place before it was set up to measure oxygen production. Allowing the test chamber to dry between trials would have helped to prevent this, but as we were short in time, we did not have time to allow it to dry completely, and paper towel was not very effective in drying it out.
The significance of this experiment is that it informs about the decomposition of toxic materials in our body. It tells us there is a maximum rate at which our body can break down these molecules, and this information allows us to know how often and in what quantities you can intake toxic materials without passing the threshold where your body can break them down, leading to permanent damage. This can help doctors in knowing what steps to take when someone consumes toxic material, based off how much they consumed and the rate at which their body will naturally be able to break it down. Furthering on this topic, one could look into how your body's enzymes break down other toxic substances, such as alcohol. The enzymes involved in the metabolism of alcohol are alcohol dehydrogenase, ADH, and aldehyde dehydrogenase, ALDH2. When these enzymes break down ethanol, they release acetaldehyde, a carcinogen, which then further breaks down into acetic acid. An experiment could be designed to look into the saturation points of ADH and ALDH2, as well as how much of the acetaldehyde fails to break down into acetic acid.